Okay, I think we're going to get started as everyone still rolls in. Good morning, everyone. Thank you very much for joining us today for this really exciting event about Actinium-225 and targeted alpha therapy from production to patient care and beyond. Before I introduce the topic and our speakers today, I wanted to take a quick moment to introduce myself. My name is Melody Greaves. I am the Manager of Strategic Initiatives at the Canadian Nuclear Isotope Council, which is a not-for-profit coalition that is 85 plus members strong and growing across the Canadian isotope supply chain. Canada has been a global leader in the production, use, research, and supply of several essential isotopes and radiopharmaceuticals for many years. And the CNIC was established to bring this leadership together and to bring all stakeholders in the isotope industry together with a mission to ensure we continue this leadership role in improving people's lives. Today, CNIC, in partnership with Actineer, is very excited to be hosting this scientific webinar about an emerging isotope that is quickly gaining a lot of attention in oncology for its significant potential in fighting cancer by providing targeted treatments for patients. Because of this potential, experts are predicting that Actinium-225 will be needed for hundreds of upcoming cutting-edge radiopharmaceutical products to treat diseases like cancer. This means that demand is increasing rapidly, but in the global supply chain, Actinium-225 is known to be one of the rarest radioisotopes. So the annual supply that we're getting from global sources is only really enough for a very limited number of patient treatments. However, with our historic leadership and expertise, Canada is very well poised to fill this supply gap, and one organization that is rising to this challenge is Actineer. Actineer is the new joint venture between CNL and ITM, both of which are long-term members of the CNIC. In this event, you will hear from representatives of Actineer about production and supply, and from some of the leaders in medicine and academia focused on the use of isotopes for targeted alpha therapy and how this can have really transformative effects for patients. We have a great lineup of speakers today, but before we get started, I want to let everyone know that there will be an opportunity to ask the speakers questions following the presentation, so please feel free to submit your questions using the chat or the Q&A feature at any time. Our first speaker will be providing some really exciting information about how Actineer is innovating to produce this isotope right here in Canada. So I would like to welcome Ram Mueller. Ram is an accomplished executive with more than 25 years of industry experience in business development and the medical isotope and radiopharmaceutical landscape. Ram is president of Actineer and the vice president, head of isotope business at Canadian Nuclear Laboratories. CNL's isotope business is currently focused on developing and scaling up its Actinium-225 program. Under Ram's leadership, this program has recently hit important technology, commercial, and corporate development milestones. Over to you, Ram.
Thank you, Melody. Thanks everyone for joining. It's an honor to be presenting here. So I'll just start off with my slide deck once I get into a slideshow mode. Okay. Thank you, Melody. I have a few disclosures and disclaimers. I'll let the team quickly go over this. Don't spend much time here. I got 10 minutes and we have an excellent lineup of speakers following me. So I'll touch upon, in my 10 minutes, I'll touch upon a little bit on the medical trends which are shaping the demand of radiopharmaceuticals and also why there's so much of excitement about radiopharmaceuticals in the last few years. I'll touch a little bit on the oncology market trajectory driving the demand. We'll talk a bit about a very high overview on radiopharmaceuticals and targeted alpha therapy and how this targeting is really deployed in form of radiopharmaceutical. We'll talk about the Actinium-225 supply and demand trajectory and also why actinium has thus far been a very hard to produce isotope. And I'll have the chance to introduce briefly the Actineer, as Melody mentioned, the joint venture between CNL and ITM. So yes, to start off, the trends, there are remarkable trends in the cancer management and the treatment paradigm, which have been noted in the last 10 years, that is really driving the demand for radiopharmaceuticals and also the medical isotopes. One of the key trend is the fact that there's been a broad adoption increasing broad adoption of radiopharmaceuticals. That is, when and where the radiopharmaceuticals were not previously being used, today they are being used or trialed in the clinical space, driving the demand. There's also an advent of a concept called theranostics approach, as many of you know, which is basically the iterative methodology where you treat the disease, you treat what you see, and you see what you treat, and the better visualization, which has been enabled by PET and SPECT imaging, allows the doctors to really visualize the disease state and treat it, and then do this iteration multiple times to see that the effective treatment is being administered. Also, one of the key aspects is there are more disease-specific targeting, which is enabled by biochemical engineering in the last 15 years, driving the demand. That is, the disease markers, which are basically expressing on the disease. Those markers are being targeted by bioengineering molecules, which are labeled with therapeutic or diagnostic isotope, and enable a precise and a targeted treatment. Also, the diagnostic tools have become more powerful. There are more PET drugs today, better cameras, better visualization of the disease, and also the ability to do hybrid imaging. Last but not the least, our talk's going to focus more on this today, the emergence of alpha therapies and alpha isotopes like Actinium-225. A little bit on the global oncology market as we see it today, and the projection in the next 6 years. Depending on which market report we pick up and read, and there'll be some variance there, we believe the global cancer therapy market is close to about $225 billion today, of which the radiopharmaceutical slice of the pie is about $6 billion. So when we project out for 6 years ahead, the reports and the market intel says the global cancer market is growing. People are getting older and sicker in terms of this disease, and the projected market size would be about $320 billion. But what I would like to point out here is the projection, the corresponding projection for radiopharmaceuticals goes from $6 billion today to $11 billion, which basically indicates that radiopharmaceuticals is becoming more and more relevant in cancer treatment than in the past. You can see the relative increase in the radiopharmaceuticals growth. It's about 83% over 6 years, and the cancer market grows at 42%. A brief overview on what radiopharmaceuticals are at a very high level, what's the advantage of alpha particles, and a very brief overview on how alpha rays help treat cancer. On the left side, we are seeing what a pictorial description of a very simple pictorial description of radiopharmaceutical, typical radiopharmaceutical, where on the left side we see the radioactive compound or the radioactive isotope, which is linked literally with a chain-like chemical to the targeting molecule on the which is green, depicted in green, and this is literally like a chemical Lego put together through biochemical methods, bioengineering methods. And on the right side is we depict a cancer cell pictorially, and what we see with the red Y-shaped pictogram is basically what most disease cells will have by disease biomarkers, which express themselves on those cancer diseases. And these molecules on the left side selectively have bond together to those disease sites, and which helps the targeting for the drug, and basically brings the drug closer to the disease site, and the radioactive payload basically damages the DNA of the cancer cells, which is where the therapeutic effect is seen. Alphas are increasingly being used, being deployed because of their low penetration power, which means that they don't go astray in the body. If they can be targeted, they don't travel long distances. And hence, they deliver a very high linear energy transfer to the disease cells, which basically cause catastrophic damage to the cancer cells, which is a double helix DNA damage, which makes it very difficult for the cancer cells to repair themselves. And hence, the sustained treatment can be achieved. A little bit on the actinium demand, a supply and demand equation. On the left side, we see as of 2023, there are about 4 institutes, 4 sites on the world which have been producing actinium in very small quantities. CNL being one of them, USDOE being the other, the major one, and there are institutes in Germany and Russia as well who have produced actinium and sold commercially. However, we know that the supply chain out of Russia is slightly strained now, and hence, which is further putting pressure on the supply for the industry. On the right side, we see what is projected to be the demand for actinium. As we see in 2024, the small quantities of actinium are being used primarily for R&D, preclinical trials, and a very few handful of early clinical studies. As the years are coming by, starting now over the next few years, we are bound to see several other clinical trials being commenced, which will really require more quantities of actinium than we see today. And over the next 4 or 5 years, we will see potentially commercially approved drugs and a multitude of other clinical trials as well using actinium isotope drugs. So just going back here to summarize that clearly the supply is not equal to the projected demand, and hence there are many institutes in many places in the world where there's a race to really develop and produce actinium today. Actinium has been traditionally a hard isotope to produce and even more hard isotope to scale. There are multiple contributing factors for that because most of the known production routes involve rare or very hard to source or hard to handle starting material. The most popular route of synthesis today is the thorium-229 decay route, which has a natural decay to actinium. And the starting material for that is uranium-233, which is rare, restricted, and also a strategic and sensitive material, which makes it very hard to acquire through a supply chain like a traditional raw material. The other known route is the P2N route and the gamma N routes, which involve radium-226 as a starting material, which is also fairly hard to source through a established supply chain in enough quantities, and also fairly hard to handle and store because of the radium-226 and the radon aspect of radium-226. The routes which do not involve this complex material or complex supply chain or sensitive material are also somewhat restrictive. The thorium-232 spallation route, which is known to produce actinium successfully, actually requires a scarce infrastructure in that very high energy cyclotron of 200 mega electron volts or more is required, and there are not many in the world with this infrastructure. And if that infrastructure is deployed, the actinium which is produced out of that has high levels of undesired radionuclidic impurities in actinium-227. Any measures to mitigate these actinium-227 levels are even more difficult to accomplish, wherein the energy of the cyclotron required to do that is actually double the energy, 500 MeV cyclotron. Currently, there's only one in the world, and that deploying that comes at the cost of the capacity or the yield, which basically means we don't directly produce actinium-225. We have to produce 225 and really use that decay of 225 from the spallation target. So it comes at the cost of the yield. And the reactor-based actinium routes are less mature and have not demonstrated the technology or scale readiness. So So this brings us to the key part of the presentation. Is I'm glad to introduce Actineer to this team here. Actineer, as you can see here, is a joint venture created by CNL and ITM. As you know, CNL is a Canadian entity, and ITM is a German entity. Both companies have a very rich history in terms of radioisotopes. ITM is also very well known in the radiopharmaceutical space. These two companies have come together and put together this joint venture named Actineer, with an immediate plan of producing Actinium-225 using the P2N route. And basically, these two companies are pulling in very complementary expertise, respectively. ITM has significant know-how in the P2N technology. CNL as well has been working towards this for last several years. ITM has also gained over these years R&D experience, which is coming into this joint venture on radium purification methods and extraction methods, very, very complementary to what CNL has been working, and both companies are really jointly working towards this P2N route. In addition to that, ITM brings infrastructure as well as isotope leadership with the lutetium experience and the capability to produce and distribute actinium, as well as they are one of the key suppliers of actinium today. CNL has a broadly licensed facility with scale expertise in production and logistics for using the thorium-229. And as we mentioned, we are currently one of 4 places in the world CNL is distributing actinium. And also a stable and a well-backed entity for waste management. ITM brings in sales and marketing experience, and CNL also brings a legacy of commercializing isotopes business. All in all, both companies are investing the seed know-how, which is very complementary in this space, to really accelerate the development of a P2N route, and also co-investing in this venture with respective infrastructure as well in GMP and CNL's existing infrastructure to produce and separate actinium using their interim source for separation of actinium. So this is being done in a phased manner. On the left side, you see the current CNL source of thorium-229 actinium generator. As we know, CNL supplies ITM today with the Actinium-225 radiochemical grade. But on the second step is where the joint venture takes hold, and there is currently a development program between both companies through the joint venture, where P2N route is being developed and getting more matured, with the goal to really significantly increase the quantities of actinium the world can have in quantities of curie quantities of 3 to 6 curies per year, with a ramp up starting this year. And target design and fabrication currently undergoing, and those targets will be radiated at the cyclotron site with our radiation partners. And those cyclotron radiated targets will come back to CNL at the interim scale, at the interim stage, and will be processed at CNL, then shipped to ITM. So this P2N route will then be, the joint venture will scale up with the commercial level as the market matures, and as the interim scale is fully taken ground, and the joint venture will put together a new facility which is planned around the 2028-2029 zone, as the interim program will prove the technology and the commercial readiness as well. So I will now hand over the meeting back to Melody. We have 2 eminent clinical experts in our field who will be talking to you about actinium and the drugs and the clinical trial aspects. So over to you, Melody.
Thank you, Ram, so much for that really informative presentation. It's certainly very exciting to hear about some of the global trends in the oncology market and how companies like Actineer are now working with global partners to address some of these supply gaps that we anticipate that we're going to see, and how that's going to be happening right here in Canada. As Ram mentioned, I'm now turning it over to our next speaker, who will be discussing more about some of the potential clinical applications of targeted alpha therapy specifically. Dr. Mike Satehe is a professor and head of the South African Nuclear Medicine Research Infrastructure and head of nuclear medicine at the University of Pretoria and Steve Biko Academic Hospital. Mike has published over 260 peer-reviewed articles and has trained several nuclear physicians in the country. He is one of the global leaders concerning the treatment of prostate cancer using targeted alpha therapy. Mike is the editor-in-chief of seminars in nuclear medicine and Mike is the editor-in-chief of seminars in nuclear medicine and an associate editor of the European Journal of Nuclear Medicine and Molecular Imaging, the Journal of Nuclear Medicine, and the World Journal of Nuclear Medicine. He is the chief of the Africa Health Research Institute and holds numerous scientific honors and awards. Over to you, Mike.
Thank you so kindly, Melody, and I really appreciated that I see could invite us here. And as you know, South Africa really is actually doing a lot in this space as well via what you know, and we've been working with NTP, Nexa NTP, that is also importing and making sure that actinium is in the private market. And then, of course, the European Institute that you have mentioned, JRC, that is supporting the research, and of course, Itemba Labs as well for the generators and potential to even do the actinium as well in future. So in South Africa, we're supportive of the initiative as Actineer, and then we really are saying this is the way to go on the basis of the fact that if I could start the talk by showing that how we embrace cancer complexity and the hallmark, it's important as we speak of actinium and what we have to do, but we should also recap why we are doing that. Of course, as you can see, the complexity of it, both the macro and micro environment contributing to what actually is a problem. And therefore, we have to really make sure that we understand the cancer cell, the stroma cell, and adipocytes and immunocytes. And the next talk that would speak to combination therapies, and that, of course, will be made easier by using alpha therapies as well. But the other thing that will be interesting is the innovation part of it, not necessarily as yet out there, but I mean, it's going to be an important aspect to bring into cancer futuristically. And there are some ways in which, together with the tumor microenvironment and, of course, alpha therapies that could actually change how we look into the cancer cells. In the past, one used to speak more of infections, and it's something that we should bear in mind as well as we speak about the therapies, but not necessarily for the talk of today. Just touched on this one, would not go into the details of the different strengths of the alphas, but that indeed we must get that clinical translation correct, and we must have meetings like this that are organized by the CNIC. And then, of course, the training is going to really be critical, and of course, the guidelines to this effect. I also like showing this slide because it applies to almost every aspect of the therapies, be it beta or alphas or any other cancers that we will engage, that we have to try and understand the predictors so that we can choose our patients well by scanning them well, by obviously during the scanning, start to really speak to things like circulating tumor DNA is correlated to what we are doing. And then, of course, understand the mechanisms of resistance. And today, it can be various organs involved, but today we're speaking about the beta versus the alphas, because we're not yet in the era as yet. What is the strength and advantages of utilizing the alpha therapy? As we know that indeed the radiation is overpowered by alphas. That's really important. But again, first, the advantage is that, of course, what is underestimated, of course, clearly why we're speaking about it, the abscopal effect, inducing that immune reaction, it's really important, that immune system activation, and therefore killing the secondary untreated tumors, even if they are remote. That's the strength that really alphas does impact more than the betas can do. And then, of course, the double strand, as we know, but obviously the reactive oxygen species, and therefore that water radiolysis that make sure that the proteins, phospholipids, RNA and DNA are also destroyed. So you've got multiple streams of how you actually are tackling your cancer cells in that way. And that's really something to appreciate. But the alpha that we're speaking about today is, of course, actinium, and there is a reason why it's a lead. Ram has touched on it, but clearly, if you're going to have 4 emissions and 5 with the actinium itself, that's really a serious nano generator inside of the cancer cell that we're speaking about. You really are putting multiple emissions that are happening, but with the provision that you avoid the recoil effect, so that those daughters are not going to be running amok out of the cancer cells. I mean, for alpha emissions, it's no child's play to really let the cancer survive. But the radiobiology, dosimetry, and how we actually make sure that the radiochemistry is well done to make sure that there is a benefit of this, it's going to also help determine this. So this is a real strength that we shouldn't forget why it is like this, that we have all this other emissions that are there, as you can see. And then, of course, I will also end up by showing that indeed the gamma emissions that are there, it's an advantage that we shouldn't forget. Now we can really scan very well. I mean, the gamma emissions are scannable, and that you will actually help to display, dismiss the concerns that might be there. So that being the strength, I mean, really, it is a serious strength and therefore very potent, and therefore it's by no chance that various cancers that have already been tried in humans, as you can see, leukemia, although yes, experimental lymphoma, bladder cancer. And I have to say there's a new molecule as I speak to you now, there's patients that we actually are going to be treating with bladder cancer, but one cannot disclose what molecule it is, but labeled with actinium. So then that will be really a game changer in this space, a big game changer for the bladder cancers. The brains, you know that operations don't necessarily help much. I'll come to it, and the neuroendocrine as it is with the prostate. So in this particular instance, this is an example of the glioblastoma. As you know, usually if they're metastatic or recurring, these patients live for very few weeks indeed. But you can see with the assistance of actinium daughter substance P, which is of course intracavitatory with the help of neurosurgeons. So these patients end up, of course, going for a much longer. And then, of course, in this particular instance, you can see. My apologies, I was trying to get the windows a bit. Yes. Can you see me? I was trying to get that share screen out of the window. I got the whole thing out. Am I back now? The presentation. This bottom sharing thing, but I'll leave it nonetheless. So yes. So the other therapies that actually are going to be important because of the short path length that the actinium travels. It's going to be with, of course, with the alphas. You can't do it with the betas, and these patients often don't have a chance if really they really come back. And actinium-225 is actually promising, as you can see, and then of course it is dose dependent into the successes of which it achieves, but it is promising. Just like we've shown with this, that I mean, few weeks to 2 years, it's really a big plus, and I think very few medications can be within those cancers. But I think, as I said, the others are really experimental. If one looks into what the neuroendocrine has done, it's really good. And in this particular instance, clearly we know that there has been various somatostatin receptors, agonists and antagonists that are labeled. And then, of course, lutetium has done as well. And in the past, yttrium has done as well. But we know that indeed we have gained great strides when we utilize bismuth-213 back in the past. And then, of course, actinium currently, and even lead as well, and that is doing well. And therefore, this also, by virtue of the short path length, enables us to actually take these cases of neuroendocrine well, especially if you're going to combine them with olaparib or DNA repair mechanisms, the everolimus, and various other mechanisms, the checkpoints that are there. So this shows clearly again that indeed to achieve more than 6 years when you actually have had problems with already lutetium, as you can see in this particular instance in the German cohort from Radke Rajoville, you can see clearly that this patient really they've done well. Yes, later on they may progress, but indeed you would have taken them for years before they really come so really with a stable disease. And even with small lesions, that is what one wants to show here. In the liver, you can see the lesions that are small, they all have been eradicated. Yes, you can still re-challenge at this stage, but I mean, it's showing you that we have stopped speaking about months of saying, yes, this person has survived for some 4 months, but and the study from institution. You can see clearly again, they've demonstrated in this featured article that indeed they can treat these patients very well and they remain free for 48 months. I mean, this is remarkable if you look into this. And then again, it's the strength of Actinium that will do this. But of course, we have to balance the argument and the debate. Yes, as it does this, it's going to be something for Actineer also to look into, and the whole world in any way that lead is coming up here. And it's showing the efficacy that is there, and that's something to look into. But of course, both actinium and lead, something that is of interest. Of course, we don't have those nauseas and the sickness that you find with chemotherapy, but alopecia is a bit of an interesting side effects that we actually are looking into. Maybe some of the postdocs that are in the meeting, there are some, it might be an interesting thing to actually evaluate further as to what is happening. We know the others are there, but the alopecia, it's something to evaluate. The alpha therapies, we have treated some with the Actinium-225. Not, we didn't appreciate so much, but some centers are reporting alopecia. So then stepping out of the neuroendocrine, and I think it is the best way to, given the short time, to demonstrate the efficacy using prostate because of the large burden of disease, of course, that the prostate does. You can see clearly that, I mean, indeed, in this case, when we have surgically removed the lesion, usually they stay long, but as soon as they come back as advanced metastatic disease, really we play a role. And we have demonstrated it with beta emitters and with actinium, as it is with the first case from Tracheoville. After lutetium, clearly it goes better. And then, of course, the gains of Lancet and the therapy are to be there. But I really like showing this because to say, I think the alphas are going to be a key into making sure that we are in the correct metrics. We've got to strive for a right metric that is here. This metrics working in that going up, it's the good quality of life and increased overall survival. So we must increase the years, but as we increase the years, we mustn't cause them side effects. So we want to be here more years and then less side effects, good quality of life. So to strive to be here, it's a question of the next talk because it's a question of combination therapies. But the combination therapies, I think they are good with the alphas because the standard of care alone really is there, but it doesn't increase the overall survival. And then, of course, the alphas are great in terms of the number of years, but we have to balance the side effects that we are still encountering to really be in the correct metrics. And this is possible with this. And this is an example of, again, showing what is happening in terms of the long survival that are there, that is being in several meta-analysis and the world experience showing this. After several therapies, you still have got the efficacy that is there, the anti-tumor effect is realized and is still much better. But then I also like this study, and my colleagues in the meeting, that I think this work from Heidelberg, again, it's important to show that using actinium alone is good. You do get a really good PSA response. And of course, it will be more than you get with, for instance, lutetium, which is a beta. But then if you combine beta and alpha, it would appear there might be a similar or at least better off. So it's something to balance off. And then again, speaking to the salivary glands toxicities that are there, that are really something to remember, that the cocktail, actinium lutetium cocktail, might be a way to go. And this is demonstrated here that there is excellent response without necessarily having patients having marked xerostomia. So something to really look into, because the overall survival is actually getting better. But we have seen from Sato's work in the ESMO that, I mean, PSMA4 is good, meaning that they're starting early, but really the complete responses are great if you treat early with the betas, but it can only be better when you treat with the as well. We have shown in our work of the chemo-naive, the de novo metastatic hormone sensitive, and the post-ADT, that indeed in this curve here, which was our featured article, that really goes even longer in this particular instance. So the work of demonstrating this with lutetium is actually something that we have seen in our setup. We tend to have these patients that come earlier because either they don't want chemo or they are not having access to, and then, or they are already extensively metastasized, or their other kidneys and so on are not working. So now there are combined metastases, which I would like maybe spend some minutes on it. So you can see clearly that looking at metastasis combined to the study that we did with a lot of esteemed colleagues across the world, that we have seen that we're showing that the biochemical response, it's really great, it's replicable, 72 when you look at it, and then the PFS, progression-free survival, it's around 8 months compared to the median, which is 9. And then you can see that indeed the overall survival is 15.5, which is actually great. And when you actually come together, big data, it's important, as it was shown in this work of the Warmth Act that we have done with multi-centers. And that shows clearly that, in fact, you can have a much better prediction of what is happening. And xerostomia is known, but it's usually around 90, sometimes 80 is variable to various places. Anemia is not an issue here, grade 3 only 13%, and the white blood cell is also not an issue, 4%, and so is the renal toxicity, which is 5%. And if you look at the analysis, again, they show that indeed it's more or less comparable, but some don't have the data. So this is actually important, and we're demonstrating it to say that in what is today the largest study of actinium PSMA on patients that they have advanced prostate cancer, we are seeing that we are providing the strongest evidence of the anti-tumor effect, and it's relatively safe, save for the xerostomia, but there are other matters that you should look into that decreases the survival, such as the presence of anemia during the treatment, the pattern of metastasis in the peritoneum, and then of course the liver that might be there, and the other previous cycles that have been there. And perhaps we have to really look into this. And this, what I've shown, the survival and the PFS, but however, if you are having good responders, those tend to actually be living 2 years and above. So really picking them well, as I've shown in the beginning, that predict who's going to be a succeeder. Therefore, if your patient, for instance, anemia, you actually leading to better survivability. If your patient are showing that the PSA decline is more than 50, you have 2 years compared to those that don't show that. And if your patient have received extensive chemo, that might also be reduced. They become to be less than 2 years. And then also the number of cycles of lutetium tend to also, Melody, I'll be fast. So they tend to also show that really indeed you do have a reduction in that particular instance. So really, again, radiobiology has to come into effect to understand what is happening and then how many cycles could be there. As I have shown that combination from Heidelberg, that might be something to actually evaluate as to whether that won't reduce or increase, that they still stay to be 18 months. And then, of course, we shouldn't forget that actinium is also, I think, the key to actually unlocking the antibodies back again. The study of Tagawa for the J591 is good in a sense that you really have got reasonable overall survival without hematotoxicity, and of course without having extensive xerostomia. So indeed, the half-life of 10 days might be a very good balance for actinium, hence making it a lead molecule. This will be spoken to hopefully in the next talk, but just to show that in the pre-study with the overall survival of 11 months, this is theoretically going to be better because, as I have spoken about, those issues that we can actually promote immunogenicity. Targeted alpha therapy does that better. So now combined with the pembrolizumab, for instance, immunotherapy, that will be great. And achieved. So, and similarly, Caroline, I mean, as you talk to it, I think if you're going to give the patient enzalutamide to enhance the cells that are seen, after seeing them, then you can actually destroy. You can see less cells were seen after enza. They were seen in this study by IMED, but then we should then in that way, the alpha therapies do better. And similarly with the DNA repair mechanism, I think it will be much enhance feedback to making that the double strand test will destroy it, and an olaparib is preventing the repair, that the cells will actually take long before they can. So the positive feedback will be an extensive one. But similarly, this featured article this month on lead makes us to also think, how can we work with actinium and lead as well? I mean, as others are saying in the editorial, that is it happening? That's an interesting thing, of course, to look into lead with, of course, the other daughters that it gives out, but here it shows that you can actually image lead very well, and that's going to be an interesting aspect going forward, because in the past there were dosimetry issues with lead PSMA. It would appear that lead PSMA now can be there, and a challenge, which is of course it's needed, a challenge, and then how do we balance the two going forward. But then to balance off and finish, this is actinium PSMA that shows that we can now image it, which is important for radiobiology and dosimetry. You can see that the PET versus the SPECT is good, and then indeed the kidneys are not necessarily much a problem if, of course, there's internalization of the lesions. You can see that the lesions are still taking more compared to the kidneys. That is an issue. So this is comforting that at least it would appear with the image-based dosimetry that there might be less concern, but we still have to see on a long term. So my last slide is to say that really we have demonstrated extensive and good responses on various types of being affected, lung, brain, super scans, and then that indeed I think we really cannot afford a sustainable disease control rate without PSMA-based alpha therapy, but of course not alone, and hence there has to be a combination therapy. But alphas, I think, are the future. Thanks, Chair.
Thank you very much, Mike, for that really informative presentation. Just a reminder to everyone that there will be a few minutes to ask some questions to the speakers at the end of the presentation. So please feel free to put your questions in the chat or use the Q&A feature at any time. I'd like to introduce our last speaker today. Dr. Carolina de Aguiar Ferreira is an assistant professor in the departments of pharmacology and toxicology and biomedical engineering at Michigan State University. Dr. Ferreira's research focuses on using radio-labeled compounds for the detection of disease, cancer therapy, and modulation of the immune system. In her work, she investigates radiopharmaceutical therapy using small molecules, antibodies, and peptides radio-labeled with alpha or beta emitters to treat cancer. Please welcome Carolina.
Thank you, Melody, for the great introduction. Thank you, the organizers, for inviting me. Thank you for all of you for being here. So, yeah, I'm an assistant professor at Michigan State right now, and I'm going to show a little bit of a project that I worked during my postdoc, and then I'm carrying it into my lab. It's not. So I think Dr. Ram already explained this a little bit, but I just wanted to give an overview of what radiopharmaceutical therapy is, because when we mention radiation therapy, we usually think about external beam radiotherapy, which is actually there's a radiation source outside, and the patient goes there and receives, gets irradiated with some source, and that requires knowledge of tumor location, and the tumor needs to be accessible, and you need to know exactly where it is, and you can only treat one tumor at one time. But with radiopharmaceutical therapy, which is actually systemic administration of radiation, you can actually treat multiple tumors at the same time, but you do require a knowledge of tumor biology. So as shown before by both of the former speakers, we do need an agent or a carrier that carries this radiation to the tumor while saving healthy tissue. And we attach radionuclide in it, and we can use it for imaging or therapy. And just a reminder that the effect of radiopharmaceutical therapy, it's because of the radiation and not the carrier. So the agent or the carrier is just basically something that is going to drive radiation to the tumor. So as we know, PSMA 617 was approved in 2022 with lutetium-177, and there's a bunch of studies with Actinium-225 right now and other isotopes. But if we look at the results with PSMA lutetium, we see that the median overall survival was of only 14 months with lutetium, and the response of patients was around 45%, meaning that more than half of the patients actually do not respond well to this PSMA lutetium. So why is that? Why more than half doesn't respond? Well, there's something about dosimetry. There's a study where they show that dosimetry actually predicts response. So in that study, patients that received less than 10 grays in the tumor actually failed to achieve good response, but the patients that received around 14 gray, they actually were more likely to respond to the PSMA. And unfortunately, right now, the way they give this PSMA is that the person has metastatic castration-resistant prostate cancer, and then they go and they get a scan, and they are positive for PSMA, and then they do not do dosimetry. They actually just give the same activity to every single patient, which is 200 millicurie. But 200 millicurie for one patient might not be the same as the 200 millicurie for the other patient. Some patient might receive less than 10 gray. So we definitely need to talk about dosimetry when we are talking about radiopharmaceutical therapy. In fact, potential causes of resistance to radiation therapy or radiopharmaceutical therapy could be both. One side is dosimetric causes, and the other side is radiobiological. So first, dosimetric causes is basically there is a low dose deposition in the tumor, right? So here on the left, you can see that one aspect of it is just insufficient uptake and retention because of low target expression. And low target expression we cannot change, right? The patient has whatever he has in the expression, so we cannot increase the expression of PSMA in their tumor cells, for example. Also, this can be caused due to heterogeneous target expression. So some parts of tumor express a lot of PSMA, some part of tumors don't. And we cannot change that in the patient as well. Also, radioligand excretion, this we might be able to change and tune the pharmacokinetics of the agent so we can have a higher dose deposition. And also, we are concerned that because of the radiation toxicity to normal tissues, we cannot give the amount of dose that the patient needs. So how can we fix that? If we cannot change how the patient expresses and how the tumor is, maybe we can change how we deposit the dose. And we can do that using alpha emitters. So the other presenters mentioned this before, but there are key differences between alpha and beta emission. So alpha particles, for example, they have much higher linear transfer, energy transfer. So they deposit more radiation dose per decay. They also, as you can see here in the DNA, they cause the ionization density is much higher for alpha particles in a smaller path, meaning that they can promote more DNA damage in a localized manner, which is really good for micrometastases, and this can save the healthy tissue surrounding it. Also, fewer atoms are necessary to impart a given amount of energy. So for example, if you look here, the disintegration energy of lutetium is around 0.5 MeV. But if you look at the actinium, it's almost 1,000 times higher. And if you look here at the decays of some of the alpha that we have right now, we can see that for actinium, it's what Dr. Mike said before. It basically is like a in vivo generator of alpha. So all the daughters, they also decay and they release alpha daughters too. And then this can lead to a much higher dose deposition than beta, for example, or than some of the other alpha emitters. So they have shown this in PSMA patients, and they actually were able to use targeted alpha therapy, and it did overcome some of the limitations with the lutetium. For example, the response was in 70% of the patients instead of just 45, and they saw that there was no high-grade bone marrow toxicity, which was the first concern. But we still see that the dose that we can give is kept because of the salivary gland toxicity. But usually we do think of radiation, and then we just try to increase the amount of dose in the tumor. But can a tumor be overdosed? Like, do we even care about the immune system? I think the idea in the field that we just need to increase the dose and increase the dose and increase the dose, we actually forget about the immune system in this, because there are a bunch of other radiobiological causes of resistance to radiation. Some of them we can fix with alpha, changing alpha from beta, for example, tumor hypoxia, alpha, the DNA damage, we do not need oxygen or for it. You can straight promote double strand breaks in the DNA without having oxygen in the tumor. Also, some cells have enhanced the DNA repair. So even if you increase the dose a lot, it might not be enough, and you might need to promote double strand DNA breaks, then single strand DNA breaks. And that's why now they're combining beta with PARP so that it promotes double strand DNA breaks. But with actinium or an alpha emitter, we can promote double strand DNA breaks directly. And also, a big part of radio resistance is the modulation of the tumor microenvironment. And the way we are thinking right now, we are actually forgetting about this. And we know from external beam radiation therapy and some studies right now with radiopharmaceutical therapy that radiation actually modulates the tumor microenvironment. So in the beginning, there is a cell death and DNA damage, and there's a cytokine and chemokine release, and then there is temporary depletion of lymphocytes or immune cells, but then it promotes antigen presentation, and then it can enhance infiltration of inflammatory cells. It can promote immunogenic cell death and antigen release. We know this with radiation therapy, external beam, but this is impractical in metastatic scenarios, which is mostly why we're going to use radiopharmaceutical in the first place. But all this, there's not many studies in. Does dose matter? Does the emission type matter? The tumor type, the timing? All this can be affected depending on how we are using it. So this is what the main focus of my lab is, like figuring out how we can just basically understanding what radiation does to the tumor, and most specifically to the tumor immune microenvironment. So we have in the lab a pan-cancer targeting agent called NM600. It's a APC analog. It basically just go inside the cells through lipid rafts, and these lipid rafts are overexpressed in basically all types of cancer. So we've seen active accumulation and specific tumor accumulation in a diverse range of tumors. But we wanted to actually check what radiation does to the immune system. So in this first paper published in Science Translational Medicine, we saw that actually low dose of beta increased inflammatory cytokines and it enhanced effector T cells, which are actually the ones that promote tumor death. So then we wanted to see, we compared effects of lutetium and yttrium-90, which are also beta. And we did see that there is increased in pro-inflammatory cytokines, increased in infiltration of CD8 cells, but we saw that the CD8 cells that were infiltrated were more on this dysfunction or exhausted state. So even though we did see CD8 presence, there was actually no change in tumor response. And we also saw that Tregs did not lose the suppressive function. And when we combined beta therapy with immunotherapy, it was only effective when we blocked Tregs. So there's a lot going on here. And then we wanted to like build on that and figure out the differences between lutetium and actinium, just basically figuring out the difference between beta and alpha. And to do that, we wanted to use, we wanted to do dosimetry first because we wanted to give the same dose. So if I give the same dose of beta and the same dose of alpha, is it the same? Are we getting the same response? So is dose the only thing that matters? And the answer is actually no. So when we gave our agent with the beta, we actually, these are models of prostate cancer. we actually didn't see much of a response, and there was no improved overall survival. But when we gave actinium with the same dose, we actually saw that there was much better anti-tumor response, and this was the first time we can actually control the MyCap tumors. These grow very, very fast. It was the first time we actually saw response, and as you can see here, the overall survival, the mice survived 3 or 2 or 3 times longer when with actinium when compared to the control and when compared to the lutetium. So we gave the same dose, and we just keep thinking about, oh, high linear energy and just increase the dose. But we can increase the dose with beta, but are we going to see the same effects? And the answer is no, because of the tumor microenvironment. So we wanted to figure out how or why, or potentially why we could explain that same dose, very different results. And first of all, we saw that the lutetium drives increased presence of immunosuppressive cells. So as if you can see here, the MDSCs and the Tregs, which are actually suppressive of the immune system, it actually went up with lutetium, but they went down with actinium. So in the end, a CD8 Treg ratio, which means like you have more active immune system than suppressive immune system, it went down with lutetium, meaning that the immune system there is suppressed, but it went up with actinium, meaning that the immune system there is activated. And we think it's because Tregs are usually more radio resistant than the other T cells. So if you give something with a higher linear energy or dose deposition in the Tregs actually went down, but for lutetium, that dose was not enough to promote decrease in Treg population. Another thing we saw is that with actinium, we actually saw a more active CD8 repertoire. We looked into markers of activation, memory, and proliferation. And those markers stayed very similar to the control in the lutetium group. But when we look at the actinium group, those markers significantly increase with both doses, not just the high dose, the low dose and the high dose of actinium that we gave. And this may contribute to a better anti-tumor response. We also analyzed 26 chemokines and cytokines in our tumor tissue. We saw that those changes are very dose dependent. Usually a lower dose, even with the same agent, is different than the higher dose. But we saw some interesting differences between the beta and the alpha emitter. For example, we see that IL-4, which is usually linked to poor prognosis, activation of androgen receptor, and increased proliferation prostate cancer, actually went up with the lutetium, but it went down with actinium. The same pattern was with IL-23, which is actually, it just promotes cell survival and proliferation. It went up with lutetium, but it went down with actinium. We also saw that some chemokines that drive immune activation was much higher with actinium than lutetium. We also saw something interesting, that levels of PD-1 and on CD8 cells and PD-L1 on the suppressive cells, MDSCs, actually went up with radiopharmaceutical therapy, either lutetium or actinium. And this indicates that radiopharmaceutical therapy might potentially synergize with immunotherapy, because you're increasing the PD-1 and PD-L1, and that might be better. That might promote better anti-tumor response and immune activation when you combine with immunotherapy. And people are doing that a lot, right? There are several preclinical, and there's like, I don't know, 500 clinical studies or something like that, combining radiation with immunotherapy. But the responses are contradictory. Some studies say that it's positive with beta and positive with alpha, the combination. Some studies say that it's not worth it and it doesn't matter. Or some studies said that radiation might actually make it worse. So the immunological effects were diverse and contradictory. So we definitely need more studies, specifically from basic researchers and preclinical researchers that can guide the clinical trials. And we need to look at dosimetry, treatment schedule, timing, the tumor characteristics. All this matter. It's not just a one recipe, or you use alpha and you combine with immunotherapy. It depends on the dose. It depends on the timing. It depends on which tumor. So we definitely need a collective effort from the field to elucidate those questions. And I do think that the field, we are going to shift towards radioimmune biology. We cannot separate the radiobiology from the immunobiology anymore. We need to always consider the immune system. The question, though, of alpha versus beta therapy, I think it's going to come from the top. And do we use them combined? Is that good? We just seen from the previous presenter that it might be better to just give alpha alone. But is it also tumor and dose dependent? Also, combination of therapy will depend on the type of radiation you're giving. But right now we're focusing on, DNA damage repair inhibitors, and they might synergize with radiation and immune system. But I feel like the field is forgetting a little bit about the immune system. And what about Tregs? Are they my last experiment, I thought that my conclusion was that the Tregs were actually the ones that were causing resistance to radiation. So should we focus on Tregs and try to figure out how we can block them so that radiopharmaceutical therapy will work? And also dosimetry. We need to figure out a way to personalize treatment, and for each patient, they should get a scan done and figure out, to give them 10, 15 grays, we need to give them this activity. And this those gonna go to the bone marrow and whatever, and can we give it to them? But I really think that the field is shifting towards radioimmunobiology and targeted alpha therapy, but there's still a lot that we need to know. Wanted to thank you for inviting me and paying attention to this. This is my team now at MSU. I also like to thank my team at UW Medicine. They are the ones that started me on this, and I'm gonna build my career on this.
Thank you so much, Carolina, for that really informative presentation. Recognizing that we have reached the end of our time together this morning, I just want to thank everyone for this great discussion. Unfortunately, we won't make it to the Q&A, but you have our contact information, so feel free to reach out with questions, and we're very happy to answer and to respond. Personally hearing from the experts today about how targeted alpha therapy can provide some really life-changing results for cancer patients. This is such an exciting area for both oncology and for industry, and really an exciting time for Canada as we are leading new innovation in the production of promising isotopes like Actinium-225 through new ventures like Actineer. For anyone who missed today's webinar, or if you'd like to watch any of the presentations again, the recording of the webinar will be available on the CNIC's website within a couple of days, as well as our YouTube channel. I would encourage everyone to check out the CNIC's website at canadianisotopes.ca for some more resources, some past event recordings, information on our upcoming events like the Canadian Radiotheranostics Leaders Summit that will be in June this year, and how to get more involved. You can also sign up for our monthly newsletter to be the first to hear about some of our upcoming events. Once again, thank you all for joining us today, and to Ram, Mike, and Carolina, thank you for sharing your expertise and joining us. Thanks, everyone, and we will see you at the next event.
Thank you.